In this video, we can discuss about biosensors. Here, we will discuss definition, components, working and applications of biosensors. So, what is biosensors? Biosensors is an independently integrated receptor transducer device which is capable of providing selective, quantitative or semi-quantitative analytical information using biological recognition elements. So these are the devices which will analyze the biological variations and it will convert into some electrical signal by using transducers. So it is an essentially analytical device which incorporate biological or biological derived recognition elements to detect a specific bioanalyte integrated with transducer to convert a biological signal to electrical signal. So in simply, it will bind with some biological recognition elements and it will convert the product formed during the binding of biological analyte with the recognition element and it will convert to the electrical signal by combining with a transducer. Now, what are the characteristics of an biosensor? First one is linearity. It is the maximum sensor output signal that is detected by the sensor. So it should be high for a biosensors. Second one is sensitivity. It is the magnitude of electrode response per unit substrate concentration. Another one is selectivity. Electrode response in presence of other interfering chemicals or foreign material. It should be minimum. It should be selective. It should be act against specific analyte. Then stability, it is the maximum electrode response over a period of time. It should be long lasting. And limit of detection, the lowest quantity of substance that can be determined from a biosensor. So these are the different characteristics of biosensors. Now what are the superior or sensitivity of biosensors in comparison with the physiochemical analytical instruments? As we all know, biosensors do have some specific immobilized biological materials which is in contact with appropriate transducer in order to that the biochemical signal is rapidly converted into the electrical signal. And also immobilization of biomolecules allow reutilization of such molecules. So what will happen? We can permit the simplification of full assembly of biosensors even though there it is expensive. Then biological sensing element is strategically positioned in small specific zone and is also extremely sensitive. So here we can analyze the substance even in small feasible content, and this is designed and developed in specific need-based analogy. So the biosensors are specific and broad spectrum. So these are the superior and sensitivity of biosensors when compared with the other physiochemical analytical instruments. Now, what are the components? Now, what are the underlying principles? As we mentioned before, here we are combining the biological components with a transducer device. So, there are two major parts for the biosensors. One, biological components, which is for sensing the presence as well as the concentration of analytes. And another part is transducer device, which is used to convert the physical, chemical, electrical signal to read in such way that this is the reading. Now, what are the components? First one is analyte. We have to use a substance of interest to be detected for testing or to identify some components by using biosensors. Then we have to use a bioreceptor. That is the molecule that is specifically recognize the analyte and they will react with the analytes. Now transducer, these are the element that will convert one form of the energy to another. So by combining the analyte with the bioreceptor, it may produce some physical change or chemical change or electrical change. So this change will be converted to another form of energy by using transducer. Now electronic parts, it will process transduced signal and prepare for it to display. Even though we are getting a signal, we have to make this signal in such a way that it will give a reading. So for that purpose, we will use an electronic component. And finally, an display in order to display the uh, modified or transduced signals by using interpretation systems such as liquid crystal display on computer or direct printer. Now this is the schematic diagram of an biosensors, and we have two part, biological component and physical component. First we have to use an substrate or analyte to be determined, and this is passed through an outer protective membrane. So it will not interfere or it will allow passes through the membrane, and this will bind with a sensing part, like uh, analyte will interact with this sensing part, and it will produce some changes, and or it will produce some components, and this product will passes through an inner membrane. So the inner membrane will bind passes through the inner membrane, and the product will bind with a transducer. So this transducer will convert this signal into an electronic part or microelectronic part to get a digital readout and finally to record. So now we can discuss what is the working of a biosensors. It includes four steps. In first step, the sample containing analyte is first passed through the membrane so as to eliminate the careful and most interfering molecules. So this molecule or this membrane is used to allow only the passage of analyte or substrate to be analyzed through this membrane. So this membrane will block other material that are interfering with the detection of biosensors. Now, the purified sample is then made to interact with the biological sensors to yield desired product that may represent it as appropriate chemical entity or production of heat or electric current or a charge. So that is the second step. Now, the product formed by combining the analyte with the substrate to be analyte with the biological sensors, this product is subsequently passes through another membrane or inner membrane, which will ultimately gain an access to the transducer that eventually convert the biochemical signal into corresponding electrical signal, which may be adequately amplified, read, and finally either on digital panel or recorded on suitable recording devices. So after this is the after binding with the analyte, it passes through an membrane, and it will gain access with the transducer, and it will convert the biochemical signal into an electrical signal in such a way that to display the result. Then most important thing is first step and second step is most important, because it will afford the measurement of concentration of substrate in situ without any processing whatsoever, so thereby avoiding any consumption or wastage of sample under the assay. So that is the working of biosensors. Now, what are the advantages of biosensors? These are rapid and continuous, can use for rapid and continuous measurement. This have a high specificity because the substrate which is analyzed should only bind with the specific uh, substrate or signal molecules. So the detection is very specific, and very less usage of reagent is required for the calibration, and faster response time. In short period itself we can determine the amount of analyte, and ability to measure non-polar molecule that cannot be estimated by other conventional devices. Now we can use different type of electrodes in biosensors, and most important two is, first one is biochemical electrodes and microbial electrodes. Biochemical electrodes is also known as enzyme electrodes. They are new type of detectors or biosensors that have been extensively designed for potentiometric and amperometric assay of substrate. For example, for the determination of alcohol, amino acid, glucose, and lactic acid. And another important type of electrode is microbial electrode, which is designed meticulously for estimation of glucose, lactic acid, and acid, assimilable carbohydrate, etc. And the microbial sensor essentially comprises a microorganism immobilized membrane and a pair of electrodes like lead anode, and platinum cathode. In fact, several types of sophisticated microbial electrode have been duly employed, tested, and applied skillfully to the precious measurement of biological substrate. So these are the two important type of electrodes: microbial electrodes and electrochemical electrodes. Now, what are the applications of biosensors? Biosensors can be used in different fields. Like first one is in the field of medical diagnosis, like for the determination of blood pressure, or determination of blood glucose level or lipid profile, etc. And it can also used for the monitoring program in industrial setup, which is accomplished by with the help of enzyme meticulously, so as to obtain the optimum yield of desired product. So if the desired product is there in the fermentation tank, it can be monitored using biosensors. And biosensors is based on the highly specific nature of reaction, which play a role in clinical analysis, which is used in the measurement of particular metabolite, which may serve as definite guidelines or indicator for prevailing pathogenic parameter or metabolic disturbances. Biosensors also can be used for the monitoring of various pollutants in the water. It can be used for the remote sensing of admixturing of mine gases in adverse environment. It can be used in the fermentation industry, like for the production of alcohol from molasses. And can be used for monitoring of the final desired product and cell culture, which are duly assigned by the biosensors. Biosensors will help in the monitoring of toxic gases, like in chemical industry and war. Biosensors do help in assay of concentration of ions. Respiratory gases like medicinal gases can be monitored by the biosensors. And in latest innovation, it can be used to determine the freshness of meat and fish products by inserting the biosensors directly into the food. So if there is any specific chemical entity during the putrefaction or process of decomposition, it may react with the biosensors and it will give an reading. So these are the applications of biosensors. So hope it is clear. Thank you for watching this video.